Monday Morning Joe is a quick-hitting coffee talk style 4-episode series that will cover what you need to know about antibody-drug conjugates, or ADCs, for the treatment of the historically difficult to treat triple-negative breast cancer and the endocrine therapy-resistant HR-positive HER2-negative breast cancer, now the so-called HER2-low or HER2-0 metastatic breast cancer patients. So welcome to this episode of Monday Morning Joe, and I'm Joyce O'Shaughnessy. Today we're going to discuss the targets and mechanisms of action of available and emerging antibody-drug conjugates. Please remember to subscribe to the Exchange CME YouTube channel and make sure notifications are turned on to be prompted when new episodes are released. So, breast cancer is the second most common cancer in the world, and it's a very heterogeneous disease, and it's the leading cause of cancer mortality in women. And although more than 80% of breast cancer cases are diagnosed at early stage, about 5 to 10% of patients present with de novo metastatic disease, that is, metastatic disease from the outset. And even with our best, most highly curative therapies that we have for metastatic breast cancer, still approximately 20, perhaps 30% of patients will ultimately die of their disease. Furthermore, the 5-year overall survival rate from all subtypes of metastatic breast cancer put together is approximately 30% from the time of their initial diagnosis of metastatic disease. So the first thing we got to know is what kind of metastatic breast cancer it is. And we routinely test for 3 biomarkers on all metastatic breast cancer patients: estrogen receptor, progesterone receptor, and HER2 status. These, of course, will dictate what treatment the patients receive, with endocrine therapies for HR-positive breast cancer, HER2-targeted therapies for HER2-positive breast cancer, and chemotherapy and possibly immunotherapy or PARP inhibitor for patients with triple-negative breast cancer. So let's take a look at metastatic triple-negative breast cancer. Unfortunately, a very aggressive subtype of metastatic disease with a poor outcome in spite of our best standard therapies. Triple-negative breast cancers do have a higher overall recurrence risk, a higher risk of developing distant metastasis, including brain metastasis, and the poorest and shortest survival that we have for metastatic patients. And pre-existing or intrinsic chemotherapy resistance is really a huge hurdle for the successful treatment of this subset of patients. By definition, of course, estrogen receptor, progesterone receptor, and HER2 are not viable targets for this group of patients. However, TROP2 is an epithelial antigen which is overexpressed in 80 to 90% of triple-negative breast cancers, and it is a definite treatment target for these patients. Well, let's talk about the classification of HER2-positive breast cancer. Historically, of course, the classic way to define HER2-positive breast cancer is via IHC and or FISH. And we do know that 3+ overexpression or HER2 gene amplification is observed in about 15 to 20% of human breast cancers, including metastatic breast cancers. And these, of course, we call HER2-positive disease. And so we've got really great therapies for patients whose breast cancers are classically HER2 positive. Historically, we've had fewer treatment options and less effective treatment options for patients whose breast cancers are classically HER2 negative. However, recently, there is a new subtype of breast cancer, HER2 low. So not classically HER2 positive, but either low expression, HER2 1+ or 2+, and FISH negative, or HER2 ultra low, which is actually HER2 0. This new subtype represents a new classification that we can target with antibody-drug conjugates. And the HER2 low population is itself very heterogeneous. It can be triple negative, it can be HR positive, it can be HER2 0, or it can be IHC 1 or 2 plus. And there are about 50% of all metastatic breast cancer cases are indeed HER2 low. About 2/3 of those that are HR positive, HER2 negative or HER2 low, and about 40% of triple negative breast cancers are HER2 low. So let's talk about antibody-drug conjugates in metastatic breast cancer. This is an enormously exciting area of research, and it is rapidly supplanting our use of classic cytotoxic agents, and that's only going to continue in the years ahead. So they're not yet recommended for first-line treatment. That's going to change in the near future. There are a number of first-line trials ongoing, but we have several ADCs. As you know, we have two that target HER2, trastuzumab and tansine, which is TDM1, and trastuzumab deruxtecan, or TDXD. We also have sacituzumab govitecan, SG, which is an ADC that targets TROP2, which is a surface biomarker that's expressed on a very large percentage of all breast cancers. And there are a number of other ADCs ongoing in clinical trials, and we have some data that we'll talk about with those. So let's spend a few minutes walking through the mechanism of action of ADCs in general, and then specifically trastuzumab deruxtecan and sacituzumab govitecan, so we can better understand how these agents work and why they may be beneficial for the appropriate patients with metastatic disease.
Antibody-Drug Conjugates, or ADCs, use selective targeting by monoclonal antibodies to deliver cytotoxic chemotherapy and other bioactive payloads to tumor cells. ADCs contain antigen-binding sites that help locate and attach to a receptor on a tumor cell and a cytotoxic drug joined to the antibody by a linker. The ADC binds a receptor on the target tumor cell, and the ADC receptor complex is then internalized into the cell through receptor-mediated endocytosis. Once inside the cell, the antibody and cytotoxic agent are separated in the lysosome, releasing the cytotoxic payload inside the tumor cell. This leads to the death of cancer cells, while minimizing the damage to the healthy cells of the patient. Targeting tumor cells via monoclonal antibodies reduces off-target toxicity and enables the use of cytotoxic agents that are much more concentrated than standard chemotherapy. One example of an ADC used in the treatment of patients with metastatic breast cancer is sacituzumab govitecan. Sacituzumab govitecan is composed of sacituzumab, a monoclonal antibody targeting trophoblast cell surface antigen 2, or TROP2, connected by a cleavable linker to the cytotoxic payload SN38, a topoisomerase I inhibitor. It has a high drug-to-antibody ratio with an average of 7 to 8 molecules of SN38 per antibody molecule. TROP2 is a surface protein frequently expressed by many cancers, including but not limited to breast, cervical, and colorectal. SN38 prevents the religation of topoisomerase I-induced single-strand DNA breaks, and the resulting DNA damage leads to apoptosis of the tumor cell. Trastuzumab deruxtecan is another ADC used for the treatment of patients with metastatic breast cancer. Trastuzumab is a monoclonal antibody which binds human epidermal growth factor receptor 2, or HER2, on tumor cells, and deruxtecan, the cytotoxic payload, is a topoisomerase I inhibitor. Deruxtecan prevents the religation of topoisomerase I-induced single-strand DNA breaks, and the resulting DNA damage leads to apoptosis of the tumor cell. As you can see, by changing the target of the monoclonal antibody and the payload, ADCs can be tailored to target specific cancer subtypes. The rapid advancements in ADC research have led to numerous approvals throughout oncology and have provided valuable options for challenging subtypes of breast cancer, such as triple-negative breast cancer.
So as you can see, ADCs can deliver their highly cytotoxic payload directly to tumor cells with reduced off-target effects of normal cells. They're very highly customizable based on the specific monoclonal antibody, the linker, and the cytotoxic payload. So let's talk about the specific indications that we have in practice today for these ADCs in metastatic breast cancer. Let's start with TDM1. Now, this is for patients who have HER2 positive breast cancer, IHC3+, and or FISH amplified, who have been previously treated with trastuzumab and a taxane separately or in combination. Patients should have received either therapy for metastatic disease or developed disease recurrence on or within 6 months of their adjuvant therapy. With regard to T-DXd, this is indicated for patients with unresectable or metastatic HER2 positive breast cancer who have received a prior anti-HER2 based regimen, either in the metastatic setting or if they received neoadjuvant or adjuvant therapy and developed disease recurrence on that therapy or within 6 months of completing that therapy, then they're also eligible now for first-line T-DXd. Also, T-DXd is indicated for patients with metastatic HER2 low breast cancer, so IHC 1 or 2 plus, FISH negative, who also have received a prior chemotherapy regimen in the metastatic setting, or again, developed disease recurrence on their adjuvant therapy or within 6 months of their adjuvant chemotherapy. So, in addition, FDA has granted priority review of a supplemental biologics license application for T-DXd for the treatment of metastatic HER2 low or ultra low, that is IHC 0 membrane staining, in patients who have received at least one endocrine therapy in the metastatic setting, that is, as first-line cytotoxic therapy. Sacituzumab govitecan is indicated for patients with unresectable, locally advanced, or metastatic triple-negative breast cancer who have received 2 or more systemic therapies, at least one of them for metastatic disease. It is indicated for patients with metastatic or locally advanced unresectable hormone receptor positive HER2 negative, that is IHC 0, 1 plus or 2 plus, but FISH negative metastatic breast cancer who have received endocrine therapy and at least 2 additional systemic therapies in the metastatic setting. So the take-home points about the ADCs are that they really have novel mechanisms of action that combine targeting the cancer cell with a monoclonal antibody and delivering a highly toxic payload of a chemotherapy agent directly to the tumor cells. Sacituzumab govitecan is indicated for patients with HER2 negative metastatic breast cancer, triple negative, or HR positive HER2 negative. Thank you for joining me today. As discussed earlier, please check back for new episodes on the Exchange CME YouTube page. Clinicians, nurses, and pharmacists can also visit exchangecme.com for free access to CME in a variety of therapeutic areas. Thanks again. We'll see you on the next episode of Monday Morning Joe.